Okay, so in this video, we're going to look at Unit 3 for CCEA. So we'll look at the practical elements and some other types of questions that have come up over the years in the past papers. So again, you can pause this at any point, rewind, etc., to go back through things. But these are the practicals. Some of these are the prescribed practicals. Some are the practicals that can come up as part of the Unit 3 exam. Okay, so again, pause that. That's in the front of your revision book, but have a look and pause that if you need to see. But I'm going to go through each of these. So I'm not going to go through all the methods. You can pause and have a look at them for yourself in terms of the text of the methods, but I'll go through pictures for each of the methods and talk through it to see how you get on. So there's the first one is aseptic techniques. So that's really looking at how we keep things sterile when using bacteria. Okay, so I'll leave that method there for a second. You can pause that. But to go through, so if you have to pour or prepare the agar yourself, you would use sterile agar into your Petri dish, lift the lid up at a 30 to 45 degree angle. So you don't lift the lid straight off because it can get contaminated. Then you use an inoculating loop. So it's just a metal loop. And we flame that first. So you put it into your Bunsen burner, and that's called flaming the loop. And that kills any bacteria which are on the metal loop to begin with. So it makes it sterile. Then you take your sample of bacteria, either in a conical flask or in a test tube like shown here. So we flame around the top of the test tube to make sure there's no bacteria around the top after you remove the lid. Then you take your sample with your inoculating loop. Okay, so just dip it in your bacteria. Then you would flame, it's not shown here, but you would flame the top of the test tube again, place your lid back on, open your Petri dish again at an angle, and you scrape on your egg or your bacteria onto the agar. Okay, and that's your sample of bacteria on the food substance or on the agar plate. Okay, and finally, again, you would flame the loop to kill any bacteria which are on the loop once you've finished. Okay, if you're doing a streak plate, as you've done this year, so you follow that method, flame your loop, take your sample, and then in terms of streaks, one streak first of all, and then another streak, another streak, another streak. The number of bacteria decreases with each streak. You can flame it in between, which gets rid of some of the bacteria as well, but that allows you to see those individual colonies of bacteria, which is what you're looking for in your streak plate. Okay, the next one to do with the immunity topic is your antimicrobial properties of plants. Again, pause the method to have a look there. But essentially with that one, you place you have a Petri dish with agar, you put bacteria on it and you spread it out. Okay, this is often the spread plate method or the lawn method it's called. So you use maybe a piece of glass to spread out your bacteria, like so. Then on top of the bacteria, you place little bits of paper or filter paper that you've cut out, which are dipped in your plant material. Okay, or you can use a straw to take out a sample of the agar and put some of your plant material like your ginger or your garlic in there, and then you leave that for a certain period of time. The clear zone here shows which is the most effective. So 4 in this one looks to have a larger clear zone. You might measure the clear zone or the diameter of the clear zone, diameter of the circle there, to see which one is most effective. Okay, the least effective one then is the one that has the smallest clear zone. Okay, other things they could ask you there, or other things moving on, is really looking at some microscope slides or electron micrographs. And with that one, they could ask you about the kidney. So here you've got your glomerulus with your Bowman's capsule around the outside. They could get you to label certain bits of these. You can see probably the proximal convoluted tubes around these. Okay. Also, proximal convoluted tubule here with its microvilli to increase the surface area. You have lots of mitochondria here for producing ATP for active transport. you have the invaginations down at the bottom, you have your blood supply close by. Okay, so remember that's for selective reabsorption. So mainly all of the glucose, some water, etc., absorbed in through there. Some by diffusion and some by active transport. Okay, lots of mitochondria. That's just your nucleus that you can see. So you have individual cells there, which you can't really see the cell membrane of. Okay, again with electromicrographs, you got your nerves. So here you would see, or here you can see myelinated neurons, so swan cells wrapped around to give that myelin sheath. You've got the axon inside here of the neuron as well. You can see these are smaller neurons. They could give you some which are myelinated, some which are not myelinated. You could maybe have to decide which moves faster. So thinking of saltatory conduction, that type of thing for that. The diameter of the axon will increase the speed of transmission as well, if you remember. Excuse me. Other ones then to do with nerves are your synapses. Okay, so you would know that this is the presynaptic neuron because it's got the synaptic vesicles, so the neurotransmitter vesicles, which then diffuse or move to the synaptic cleft. Okay, receptors on the other side. They could ask you here why you don't see the axon, and that's just it's in a different plane, so it's maybe behind where the pictures or where the sample has been specimen has been taken from. Again, there is one, I think, just out of the textbook. So your synaptic vesicles with your neurotransmitter, synaptic cleft. Again, your axon may be slightly behind or at a different plane. Mitochondria there to resynthesize your neurotransmitter. Muscle is another one where they could use electron micrographs. So you can see here the Z lines. Okay, so that's within the center of your actin filaments. The M lines, the thicker ones, you can see there in the middle of your myosin filaments, and the H zone there is just where you get the overlap between the two. So one sarcomere length would be from Z line to Z line. They could have a little scale bar here. They could ask you to work out magnification, possibly. So bring a ruler with you in your exam. So thinking about that MIA, magnification is the image size divided by the actual size. Obviously, you need to convert your units to micrometers or nanometers or whatever it is to know how to do that. Okay, types of muscle they could give you as well. So intercalated discs is your cardiac muscle. So these types of things here that you can see, again, lots of mitochondria there. You could have your smooth muscle with your spindle-shaped cells, so lots of nuclei there, or you could have your skeletal muscle, okay, which is again multinucleate, striated, similar to the cardiac muscle, but it's in these long myofibrils or microfibrils that you can see there, okay. Then another electromicrograph they could ask you is about the eye, so the retina mainly you're looking at there. So thinking about your bipolar cells, your ganglion cells above, and then your photoreceptor cells down the bottom, so your rods and cones. There's your choroid layer, or perhaps your scleral layer, depending on what they show there. That's more likely the choroid, the dark layer. So remember your rods and cones are here. So light has to pass through those ganglion cells, through those bipolar cells to get to those rods and cones. So light can be distorted slightly there or maybe blocked in certain ways. So those are just nuclei in terms of Y. Okay, those are just the nuclei of your photoreceptor cells and your bipolar cells. The next bit then is looking at populations and communities. So really looking at your hemocytometer. So they will tell you what type of square it is. Generally, they are B squares that they will give you. Okay, we'll look at one of those in a second. What you need to remember is that a B square is 0.2 by 0.2 millimeters. Okay, so it's an area of 0.04 millimeters squared. There's a 0.1 millimeter gap, which they tend to tell you in questions as well, between the bottom of your cover slip and the top of your hemocytometer slide. So that allows you to work out a volume. Okay, so it'll be 0.04 times 0.1. Okay, so the volume of one of these squares, including that 0.1 millimeter bit, is 0.004 millimeters cubed. Okay, so if they give you a sample like this that you have to count, they'll tell you the B squares. It's a B square you can see there. When you're counting, you count things on the north, so the top, and the west. Okay, so these you can see you would count. You don't count things on the east or the south. Okay, so when you're counting, just the north and the west, if any samples appear there. So if we were counting this one, you can see there's one, and I would mark them off as you count them. So 1, 2, 3, etc., 4, 5, 6, 7, 8, 9, 10, 11. Sorry, not that one. 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. I must have missed one there because it's 22. So 22 in 0.04 millimeters cubed. Okay, so you multiply 22 by 1 over 0.04 millimeters cubed, or you can just do 22 divided by 0.004, and that gives you 210 in 1 millimeter cubed. Okay, so the calculations you can see there, if they give you a sample like this, so they've counted 5 squares, you take an average of those, and then you multiply that, or you divide that by 0.004, or I think you multiply it by 250, something like that, would give you that amount of cells in 1 millimeter cubed. They tend to ask for the number of cells in 1 millimeter cubed. The other one they could ask you is the capture-recapture technique. Okay, and that is you take the number in your first capture that have been caught, your first sample, multiplied by the number in the second sample, divided by the number that have been recaptured. So if you remember, when you take your first sample, you mark those, okay, then you put them back into the population, allow them to reintegrate maybe a day later, depending on the type of organism it is. Then you take a second sample, the number that are marked in the second sample or the number that are recaptured. But that's the formula you'll need to remember. So sample 1 times sample 2 divided by the number recaptured. They sometimes ask about the assumptions here with this as well. So it needs to be a large sample which have been caught or trapped, and they need to be marked in a way that doesn't make them obvious to predators. So semi-permanent marker, bird ringing, etc. Put it on the underside of their body, etc. So it shouldn't make them more obvious to predators. You don't put a big tip X mark on their back so that predators can see it. You need to make sure that they're released back into the population and allowed sufficient time to mix. Depending on the organisms, that will not be left for a month or 2 months. It might be a day or a couple of days, depending on what it is. And then you would resample. Okay, so the assumptions here, which aren't really what you can imagine or what will happen, is that there's no significant gains through immigration or emigration. That's why you don't leave them for that long. No significant gains through births or deaths. Again, don't leave them for that long. The trapping process shouldn't affect the animal in any way, and the marked animals should have sufficient time to reintegrate back into the population. So as it says at the bottom here, it tends to be small, mobile animals that are easily trapped. Okay, trapping, you could use a pitfall trap, you could use a sweep net possibly as well. Okay, then you have the respirometer to calculate RQ. So with this one, this is a simple respirometer. So if you think you have your respiring organisms in the tube, okay, you tend to leave them for 5-10 minutes to acclimatize. Then this is a closed system. So your tap is closed. There's a certain amount of air within this system up to your bead of colored liquid here. Okay, so when you start it, the respiring organisms will take in the oxygen. We would use potassium hydroxide first here, which absorbs the CO2. So the amount of air within the tube should decrease. So therefore the bead should move towards your tube. Okay, and if you leave that for a certain period of time, then that will calculate, or that will give you the volume of oxygen consumed by your respiring organisms. Okay, to work out the CO2, you would do exactly the same, but you replace the potassium hydroxide with water. Okay, so in other words, then the CO2 is not absorbed. Okay, so the CO2 stays within the closed system. So again, you let it run same period of time as you do with the first one. If the bubble stays where it is, then the same amount of carbon dioxide is produced, as oxygen consumed. Okay, so that would give you an RQ of 1, which would be carbohydrates. If the bubble moves towards your tube, there's less CO2 produced, okay, because again, the same amount of oxygen would get used in that time. But if there's less CO2 produced, the bubble will move this way, which tends to show it's either fats or proteins being used for respiration. If the bubble moves away from the tube, then there's more carbon dioxide produced than oxygen consumed. Less oxygen, that would suggest some anaerobic respiration as well as aerobic respiration. Okay, so it stays where it is. RQ is equal to 1, so same oxygen consumed as carbon dioxide produced, moves back. There's less carbon dioxide, moves away from the tube, or in the other direction, there's more carbon dioxide. Again, they could give you this type of thing, which is exactly the same living material here. One thing to note is if they're using plant material, you need to do it in the darkness, okay, because photosynthesis would have an effect. Okay, so if it's plant material, it needs to be done in the darkness. Okay, again here, work out, so you'd have your potassium hydroxide in here, volume of oxygen would be how much this moves up the way. Again, for you working out carbon dioxide, you need to replace this potassium hydroxide with water. Okay, if it stays the same place, it's the same volume of carbon dioxide, moves up, less carbon dioxide, moves down, there's more carbon dioxide used. Okay, the other ones are ones that we didn't do this year, but the redox indicators. So with these, go back. So with these, you have your NADs and FADs pick up your electrons and become reduced. Okay, and that can be detected by either methylene blue for respiration or DCPIP for photosynthesis. So normally methylene blue, surprisingly, is a blue color when it's oxidized. When the hydrogens produced from respiration are picked up by NAD or FAD, then your methylene blue becomes reduced, so it goes colorless. Okay, so you would use, again, they use mung beans or pea beans here. If you use boiled ones, then respiration won't occur, so it should stay blue. And you add your methylene blue, allow them to respire. If it changes colorless, as you can see in tube A and tube C here, that means that respiration has occurred. Okay, tube B and tube D are the boiled ones. Okay, so no respiration has occurred there, so the methylene blue hasn't been reduced, so it stays that blue color. Okay, so if methylene blue changes from blue to colorless, it means that you've got respiration happening or occurring. Okay, for photosynthesis, it's DCPIP. Again, you would, for this one, you need to isolate your chloroplasts. Okay, so you homogenize or mash up your lettuce leaves. You tend to send that through a centrifugation, so it separates. It might be that nucleus is taken out first, and then your chloroplasts will bullet, if you like, towards the bottom or form a mass at the bottom. Lighter ones that might be towards the top, they've sometimes asked about, would be like your ribosomes or Golgi. Those types of things would be higher up in your centrifugation tube. But you isolate your chloroplasts, you use a control tube here. So again, water with DCPIP, it shouldn't change color. So DCPIP's a dark blue color. Again, it'll go to colorless if photosynthesis is happening. So here, no change. If you have your chloroplasts with your DCPIP, it changes to a green color. Okay, that should be green rather than colorless. Changes to a green color. So that means that the light-dependent reaction has occurred. So some of your reduced or NADPs have been produced. When it's in darkness, obviously photosynthesis not occurring, it should stay that blue color. Okay, so those are the redox reactions. Photosynthesis, then they can ask you about chromatography as well. So you would mash up your plant material, take a concentrated spot on your origin line. Remember, this isn't pencil. Put it into your solvent, let it run, mark your solvent front. It should separate out into colors. So one way that you can tell the difference between the different photosynthetic pigments is by the colors, but you can also work out the RQ. Okay, so you take your center of your line, whatever it is, you take the center here, you'd measure the distance to the origin line, and then you measure the distance to the solvent front. Okay, and your RF, sorry, not RQ, RF values are always less than 1. Okay, so you divide this distance by the larger distance of your solvent front. Okay, so we could get you to maybe calculate that in some of the questions. As I say, the colors that they separate out into is another way of identifying the different pigments. Okay, then with gene technology, you've got gel electrophoresis. I'll leave that up there for a second, and you can have a look at that. I'll pause that. Essentially, here you take a sample of DNA, you split it up using restriction enzymes, which split it up into different lengths. Okay, you place some of that DNA into a little well in this gel substance. You pass an electric current across it. You have the negative cathode at this end. DNA is negatively charged because of its phosphate and the oxygens within its phosphate groups. So it will repel this negative and move towards the anode, move towards the positive anode. Okay. Smaller fragments tend to move further, larger fragments not as far. Okay, so that's the type of thing there, and you get the DNA separating out into different fragments. So there's smaller fragments moving further away from the well, and the longer bands of DNA or longer strands of the cut up DNA don't move as far. And you can see that within this little animation. So if you put your DNA samples into here, pass your electric current across it, they move towards the anode. Then you can compare different samples of DNA. So they can be used for paternity testing, can be used for at crime scenes. So these two samples are the same. So if that was found at a crime scene, this is your suspect, he was there. Okay. Also with gene technology, you have the extraction of DNA. So again, your method there. So that generally can be used plant material, kiwis, things like that. You can do it with your own spit as well. So you would treat it with lysis or treat it with your detergent, which causes the cell membranes to break down. Then you treat it with an enzyme which breaks down the histones, okay, and separates out your DNA. Put it through your centrifuge again if you have one of those, and then you should get DNA strands which sit or precipitate on the top when you add your cold alcohol. Okay. The last one really is then your leaf scrape that we did. It is essentially dissection of a small animal organ or part of a plant. So we did the leaf scrape one for that. So there's the method, reasonably simple. Scrape your leaf, taking off the upper layers of the leaf, leaving the lower epidermis. You add water there to prevent friction and the breaking of the leaf. And then when you look at those cells, that's the lower epidermis. You can see stomata cells here as well. Okay. They will not ask you specifically about a rat dissection because not everybody does that. Some schools do, but we don't. They could. That one has come up before. We have to identify the types of cells. You had to identify the stomata. More than likely, with the dissection bit, they will ask you to explain how you do it. Okay, so you would pick the one that you did. So you're regurgitating the method there. Okay. Other things that have come up is writing a bibliography, which you can see here. So with a book, they could give you some text and give the author's name and the name of the book and publisher's name, etc. To write a bibliography for a source material that you've used. First, you start with the author's name, surname, and then their first initial or initials, followed by a full stop. So you can see here, J. Harris is the author. So Harris, J, full stop. Then you have the title of the book or the journal, followed by a comma. So in this case, Enhancing Evolution, the ethical case for making people better, comma, the publisher's name, followed by a comma, Princeton University, comma, and then the year of publication, followed by a full stop. Sorry, Princeton University Press, comma, 2007, full stop. Our textbook is written by Napier, Dr. J, full stop. Biology for CCEA A2 level, comma. It's published by Color Print Education, comma, and it was published in 2007. So that's the type of thing you want to put there for a bibliography. Other things they could ask you related to respiration photosynthesis is looking at just the ultrastructure of your mitochondria. So again, electron micrographs of those types of things. Could be related to magnification again. So think of your double membranes, your cristae that you can see there as well. Okay, similar sorts of things. Or again with chloroplasts. Okay, so that's sort of synoptic in terms of A2 and AS. So being able to identify each of those, maybe work out magnification from the scale bar. So that gives you your stacks of grana or your thylakoids. You can see there your lamellar grana or interlamellar layers there as well, stroma in the middle, etc. So that's pretty much it. Going through, this is in your revision booklet as well. So you've got the methods for each of those. Again, you can pause these at different points there. So you have the aseptic techniques, your antibiotic discs, which you can use, as well as plant discs. And we've used E-strips in the past where you have low concentration of antibiotic to high concentration of antibiotics. So you'd expect a larger clear zone towards the high concentrations. Your antimicrobial properties of plants we talked about. Your hemocytometer we've talked about. Just make sure it's the B square or whatever square it is, multiplied by 0.1, and then you would do 1 over that. Your northwest rule as well, if you're counting there and calculating depth. Generally, it's number of cells per millimeter cubed. You've got the RQ we've talked about there. They could ask you how you do that, or they could talk about why you put it in darkness, that type of thing, if you're using plant material. Think about the RQs there. So carbohydrates, RQ of 1, fats, RQ of 0.7, proteins, RQ of 0.9. Okay, anything more than 1, it's aerobic plus anaerobic. Again, your redox reactions, so your respiration one with methylene blue, DCPIP one as well. So think about that. Chlorophyll emitting electrons, which are picked up by NADP, causes the DCPIP to change to colorless. Chromatography, you need to have the same solvent strength to be able to identify the pigments. You've got there the different pigments will move different distances based on their solubility. Okay, so calculating RF values there again, you can see it's less than 1. Your gel electrophoresis, your anode and cathode, so restriction enzymes, electric current flows. DNA is negatively charged, so it moves towards the anode, and it can give you, or you can look at different suspects as we've talked about. Okay, extraction of DNA, add your detergent, causes the cells to lyse. Your enzymes breaks down the histones or chromosomes. Add your cold ethanol, you get your DNA strands forming either at the top or within. And then your dissection, run the blade across. As I say, we've ran through that in terms of your leaf scrape, your grass scrape. You're looking for lower epidermal cells. You can use iodine to stain it. You might put water under the cover slip just to prevent air bubbles as well. Okay, hopefully that helps with Unit 3 for next week.